So the first portion of this class is going to focus on the structures of 4 classes of biomolecules. So we will start first with carbohydrates. We will then move on to proteins, lipids, and nucleic acids. And the information that I'm talking about in this lecture is all found in the textbook on pages 275 through 298. So carbohydrates are the single most abundant class of organic molecule that's found in nature, and they have a number of diverse functions. A primary function is to serve as energy stores, so these are primarily the function of the molecules of glycogen and starch. They also form the structural framework of nucleic acids, and we'll talk about ribose and deoxyribose that form the sugar framework of RNA and DNA. They serve as structural elements in cell walls and exoskeleton, so we'll look at chitin and how that structure imparts function to cell walls as structural elements. And they can also be linked to many proteins and lipids to form proteoglycans and glycolipids. So carbohydrates can be broken down into 3 major classes. First are the monosaccharides. These are the simple sugars. An example is glucose. This is the simplest form of a carbohydrate. Then there are oligosaccharides. So these are short chains of monosaccharides, so anywhere between 2 and 10 monomers. Disaccharides are the most abundant. So one example of this would be sucrose, which is composed of glucose and fructose, which are linked together. And then lastly, we have polysaccharides, which are basically polymers of simple monosaccharides. So anything that is longer than 11 or so monomers would be considered a polysaccharide. So all monosaccharides have the general formula of CH2ON. They all have 2 or more hydroxyl groups, and in general their names are based on the number of carbons present. So we have 3 carbons are a triose, 4 carbons tetrose, 5 carbons pentose, and 6 carbons are hexoses. So trioses are the simplest monosaccharides. So these are sugars that have 3 carbons. All monosaccharides can be grouped into one of 2 categories. So that can be either be an aldose or a ketose. And the class to which they belong is dependent on the placement of this carbonyl that's shown in pink here. So this is glyceraldehyde. This is an aldotriose. The carbonyl is at the end of the carbon chain. That makes it an aldehyde group, and then this monosaccharide is an aldose. So in contrast to aldoses, we have ketoses. In a ketose, the carbonyl carbon is at any other position other than the end of the chain. This is a ketone group, and the monosaccharide is a ketose. So another characteristic of monosaccharides is that they are chiral. And so what that means is that they have 1 or more asymmetric or chiral carbons. So by definition, chiral carbons have 4 different substituents. All monosaccharides except dihydroxyacetone have a carbonyl carbon. A chiral carbon. And the exception is dihydroxyacetone, and that's because that ketone group is at the 2nd position, and so that carbon is no longer chiral. So if we go back, what we see here is now you have a symmetrical carbon. So this end of the molecule is identical to this. So monosaccharides with chiral carbons occur in optically active forms, and we refer to these as D or L forms. In the D form of sugar, this is dextrorotatory. This D isomer, when we write the formula, has a hydroxyl group on the right side of the molecule. In contrast, the L or levorotatory isomer has the hydroxyl group on the left. Now, whether or not a sugar is classified as a D or L is based on the position of the chiral carbon that's furthest from the carbonyl. Okay, so there's only one position that determines whether or not a sugar is a D or L sugar. So there are three ways to represent the L and D forms. So first is a ball and stick model, and this shows the actual configurations of molecules. Then the next kind of representation that's often used are Fisher projection formulas. So here we see D-glyceraldehyde and then L-glyceraldehyde. And in this projection, the way that we read this is that the horizontal substituents are facing towards us, and the vertical are facing away from us. And so in a very similar manner, so here are Fisher projection formulas, and then these are perspective formulas. And the perspective formulas, the dashed wedge, so we show that here, are facing away from the reader, whereas the solid wedges are towards the reader. So each representation kind of highlights a different aspect of the molecule, and so you'll see each one of them used. Aldoses with 3 or more carbons and ketoses with 4 or more carbons all contain chiral centers. So what this means is that there are multiple stereoisomers of each molecule. So in general, there are 2 to the n stereoisomers, and n equals the number of asymmetric carbons. For example, glyceraldehyde has 1 chiral center, therefore it has 2 stereoisomers. In contrast, if you had a molecule that had 4 chiral centers, there would be 2 to the 4th stereoisomers, meaning that you would have 2 times 2 times 2 times 2, and that would give you 16 stereoisomers. Ketoses also have 1 fewer asymmetric center than do aldoses with the same number of carbons, and again that's because we lose an asymmetric carbon when that carbonyl group is moved. Now the chiral carbon, there's one chiral carbon that's of particular importance, and it is the chiral carbon that's furthest from the aldehyde or keto group. And this is important because it determines whether the molecule is a D or an L sugar. So here are some aldoses. First thing I want you to notice is that in red are the chiral carbons. An aldose with 3 carbons is an aldotriose, and so we see that here. This is D-glyceraldehyde. If we look at the aldotetroses, we see those here, so these are the 4 carbon sugars. In red, again, we have the 2 chiral carbons, and the chiral carbon that is furthest from this carbonyl is the one that designates the L or D. These are all D sugars, and we have the hydroxyl group on the right. So 2 things I want to mention. One is that the sugars or the monosaccharides that are boxed are what you will be expected to know for the exam. And also in the figures, and I believe in the book, all of the sugars that structures that are given are in the D form. And so in that case, if we were to have L-glyceraldehyde, the only difference would be the hydroxyl group would be on the left. So then we have aldopentoses, so these are 5 carbon sugars. Again, they're aldoses, so that carbonyl is at the first carbon. These red carbons are your chiral carbons, and again, they are all D sugars, and that this hydroxyl group is on the right. We also have your aldohexoses. These are 6 carbon sugars. They have 1, 2, 3, 4 chiral carbons. So they have 2 to the n stereo, so they would have 2 to the 4th stereoisomers, or 16 stereoisomers. So 8 would be in the D form and 8 would be in the L form. And then here are all of the D form aldohexoses. And again, I would expect you to know glucose, mannose, and galactose from this slide for the exam. Now let's look at some of the ketoses. So if you remember, dihydroxyacetone is the only monosaccharide that does not have a chiral center. So because of this, there's only a single stereoisomer. Now, if we look at the 4-carbon ketose, we see here that there's only a single carbon or a single chiral carbon, so there are only 2 stereoisomers and ketohexoses. And here all of the same rules apply. So the chiral carbons are in red, and for each chiral carbon there are 2 stereoisomers. These are all the D forms, so the hydroxyl groups are on the right-hand side of this chiral carbon that's furthest from the carbonyl, and you would be expected to know ribulose, cellulose, and fructose. So there are a number of specific terms that we use to describe stereoisomers and their relationship to each other. And so when we have stereoisomers that are mirror images of each other, we call these enantiomers. And so D-fructose and L-fructose shown here are mirror image or enantiomers of each other. And so basically the analogy to this is your left and your right hand. So these are mirror images of each other. They're not superimposable. Now you can have pairs of isomers that have opposite configurations at one or more chiral centers but are not mirror images, and these are what we call diastereomers. And so mannose and galactose are diastereomers of each other. So they differ, they differ at the C2 and the C4 positions. And again, they are not mirror images of each other. So 2 sugars that differ in configuration at only one chiral center are called epimers. And so here we see mannose and glucose. So mannose is an epimer of glucose, so it differs only at the C2 position. Glucose and galactose are also epimers of each other. In this case, they differ in their configuration around the 4th carbon. These, however, are not epimers of each other, but they're diastereomers of each other because they differ at 2 positions, the C2 and the C4. So for simplicity, we represent structures of these 5 and 6 carbon sugars as straight forms. But in aqueous solutions, they're actually more often found as cyclic structures. And this formation of rings is basically the result of a general reaction that occurs between aldehydes or ketones and alcohols to form a number of derivatives. And so what we'll see here is that for aldehyde or ketones, they're able to react 1-to-1 with an alcohol. And these form hemiacetals or hemiketals. So we see that here. So in this first step, the carbonyl forms a covalent bond with the hydroxyl group. So we have this carbonyl forms a covalent bond with this hydroxyl group. And then you get the formation of this hemiacetal or hemiketal here. Now, this hemiketal now has an extra chiral carbon. And so this carbon is now a chiral carbon, so it has 4 different substituents on it. Now, you can have a second substitution of an alcohol. And in this case, what we get are acetal and ketals. And when that hydroxyl group comes from another sugar, we have the formation of a glycosidic bond. And we'll look at that more closely in a minute. So now let's look at the cyclization of D-glucose. So here we see in the top panel, D-glucose is written as a linear form. This cyclic structure is more representative of what it looks like. In the formation of this cyclic structure, what happens is that you have this hydroxyl group from the C5, which reacts with the carbonyl group of the C1. So here we see that reaction here. Now this results in the formation of a hemiacetal, and now again we see that this C1 carbon is now chiral. So it now has 4 different substituents, and this new chiral carbon can adopt 2 different conformations. So here we have alpha D-glucopyranose, and here we have beta D-glucopyranose. And so here with the alpha, the hydroxyl group is facing downward, and in the beta, it's facing upward. So again, the C1 is the anomeric carbon. These alpha and beta stereoisomers are anomers of each other. And this conversion between alpha and beta that goes through this linear intermediate is called mutorotation. Now, this is a Haworth projection, and the way that we translate this linear representation to a Haworth transformation is that the substituents on the right side are given a downward orientation in the Haworth structure, and those on the left side are giving the upward orientation. So again, when we looked at the cyclization of D-glucose, this alpha and beta designation indicates the position of that hydroxyl group of that anomeric carbon to that of the hydroxyl group of the highest numbered asymmetric carbon on the ring. And so here we see the hydroxyl group at the anomeric carbon versus the hydroxyl group at the highest asymmetric carbon on the ring. So these are on the same side, and this is alpha D-glucopyranose. So these are cis or same, gives you the alpha configuration. If we have this hydroxyl group, which is on the opposite side of the hydroxyl group of the highest asymmetric carbon, these are in trans, and that gives you the beta form. Now the D-glucose, or the D form, is indicated by this CH2OH group. So here we see it's above the ring. If it's below the ring, that would be an L sugar. So hexoses can form 5 or 6 carbon ring structures. The 6 carbon ring structures we call pyranose, and that's because they resemble this pyrene group. And then the furanoses, shown here, resemble the furan group. So here we have aldohexose. This is glucose. So we have alpha D-glucopyranose. So this pyranose indicates it's a 6-membered ring. D means this CH2OH group is on top of the ring, and this alpha indicates this hydroxyl group is facing downward. Here with the beta D-glucopyranose, the only difference is that this hydroxyl group is facing upward. Here we can see that hexoses can also form a 5-membered ring, so this is a furanose. So again, the alpha carbon is shown here, the anomeric carbon is here. The hydroxyl group of the anomeric carbon is shown here, and it's on the same side as the hydroxyl group of the highest asymmetric carbon on the ring. So then this is alpha. Beta has the hydroxyl group in the opposite orientation. And again, these are D sugars because they have their CH2OH substituents on top of the ring. In these Haworth projections, to orient you, the bold edge is the edge nearest the reader. Aldohexoses can adopt either ring structure. So they can be pyranoses or furanoses, but in general, aldopyranoses are more stable. The other thing to point out is that C1 is the anomeric carbon for aldopyranose. And so we see here, this is the anomeric carbon. And for furanoses, C2 is the anomeric carbon. So this is what is going to determine your alpha or beta positions. Here we see, again, this is another representation of the cyclization of D-glucose. And we see here that it can form a 6-membered or pyranose group ring or a furanose ring. And the type of ring that is produced is determined by which hydroxyl group attacks the aldehyde. So if we look in blue arrows, so we see here the C5 hydroxyl attacks the aldehyde, then we get the formation of the structure highlighted in blue here, which is the pyranose form. If the C4, you see that in red, if the C4 hydroxyl group attacks the aldehyde, you get this furanose form. So again, these hexoses can, or glucose can form either a pyran group or a furan group. So here is the cyclization of D-ribose. And again, we can get the formation of 6-ring structures or 5-ring structures. And that is determined on the basis of which of these hydroxyl groups attacks the aldehyde group. Here we see that the hydroxyl group, so this is the alpha anomer, this is the beta anomer, alpha and beta. So again, you can form either ring structures depending on which hydroxyl group attacks the carbonyl group. Now what's kind of interesting is that in solution, almost 80% of ribose exists as a pyranose. So it's almost all found in this 6-membered ring. However, when we look at the structure of nucleic acids in RNA and DNA, all of the molecules, all of ribose is found as a 5-membered ring. And so that's because the stability is due to other atoms that are around that molecular composition. So now let's look at the cyclization of D-ribose. So here we see the linear structure. We see here that it can form a 6-membered ring or a 5-membered ring. If the C5 hydroxyl is the one that attacks the C1 of the aldehyde group, we get the formation of a 6-membered ring. And we see here we have alpha D-ribopyranose. So pyranose is the 6-membered ring. And we see that alpha is shown here with this hydroxyl group pointing in the downward position. You can also, the other anomer of that would be beta D-ribopyranose with the hydroxyl group pointing up. If you have the C4 hydroxyl group that attacks the C1 aldehyde, you get the formation of a furanose. So now we have this 5-membered ring. The alpha has the hydroxyl group pointing down, and the beta has the hydroxyl group pointing up. Now what's kind of interesting is if in solution, about 80% of ribose exists as a pyranose, so this 6-membered ring. However, when we look at the structure of RNA and DNA, the ribose is found exclusively as a furanose. So in general, aldoses and ketoses that have 5 or more carbons form either a furanose or a pyranose. Aldohexoses, because of their structure, tend to prefer pyranose rings, whereas ketohexoses prefer furanose rings. Now, we've been drawing these cyclic structures as Haworth projections, and that's because they're easy to visualize. However, these structures are not planar. and monosaccharides tend to adopt one or two different conformations. So here we see the Haworth projection. Here we see a chair conformation, which is a more accurate representation of the way the molecule exists. And then we also, monosaccharides can be found in a boat conformation. Now, conformations, so this boat form is found only when you have very bulky substituents. So most monosaccharides are found in the chair conformation. Now, conformations of molecules are interconvertible without breakage of covalent bonds. And so these two molecules can, or this molecule can go back and forth between two structures, and it doesn't require any breaking of bonds. And so these are conformations of each other. Now, there are different chair conformations of monosaccharides. So we have two possible chair forms. Each position can adopt either an axial or an equatorial position. So axial is parallel to the axis here, and then equatorial is perpendicular. In general, bulky substituents tend to favor equatorial positions because they're less sterically hindered. And so here we see alpha D-glucopyranose in the chair conformation. So there are a number of different monosaccharide derivatives, and we're going to talk about 6 of them. So we'll talk about sugar phosphates, deoxy sugars, amino sugars, sugar alcohols, sugar acids, and ascorbic acid. I'm not going to ask you to memorize these different molecules, but you should be able to recognize derivative types. So you should be able to tell me that something is a sugar phosphate. So here are a couple of examples of sugar phosphates. They're also called sugar esters. They are monosaccharides in metabolic pathways. And we'll talk about a lot of that in the future when we look at metabolism. Covered basic monosaccharide structure, and now we're going to briefly discuss sugar derivatives. So basically, sugar derivatives are modified sugars that function in many biological processes. So the modifications give the molecules different biochemical properties, which in turn influence their function. So here are some sugar phosphates, which is the first group that we'll talk about. So this is alpha D-glucose 1-phosphate. So this is the first step in glycolysis. The glucose gets phosphorylated, and that traps it within the cell. Alpha D-fructose 1,6-bisphosphate. Here we see the phosphate groups shown in pink. This is also a metabolic intermediate of glycolysis. And then here we see adenosine 5' triphosphate. And of course this is ATP, which we're all familiar with, that provides energy to drive chemical reactions in cells. So a lot of times these molecules are called sugar esters for the bond which links the phosphate group to the sugars. These are often monosaccharides in metabolic pathways are often converted to phosphate esters, and we'll talk about that at the end of the semester when we look at metabolism. So one thing to note is that sugar phosphates are relatively stable at neutral pHs, and they bear a negative charge. So here we can see that all of these phosphate groups are negatively charged, which of course has a big influence on their biochemical properties. So deoxy sugars are another group of sugars. What we see here is beta-2-deoxy D-ribose. This is found in DNA. And also we have alpha-L-fucose, which is also 6-deoxy-L-galactose. And what we see here is that at the 6th position, here we are lacking in oxygen, and then here at the 2 position we're lacking in oxygen. And so in all of these, the hydrogen replaces the hydroxyl group in the parent polysaccharide. So again, this is the predominant sugar in DNA, and fucose is interesting. It is a component of lots of glycans and glycolipids that are made in mammalian cells, and it is one of the few L sugars that are found in nature. So most of the sugars that we see are D forms. So next we have amino sugars, and these contain an amino group in place of a hydroxyl group. So we see here the amine group at the C2 position, and then here another amino group. So this is beta-D-galactosamine and beta-D-manosamine. So these are amino derivatives of galactose and mannose. So next we have sugar alcohols. So in this class of molecules, the carbonyl group of the parent monosaccharides, which would be shown here in glycerol, here in myoinositol, and then here in ribitol, have been reduced, and they are now producing or now resulting in a polyhydroxyl alcohol. So we usually name these by replacing "-ose" with etol. So myoinositol is actually a derivative of glucose, and D-ribitol is a derivative of ribose. Glycerol is a very important constitutive of lipids, and we'll see that it forms the backbone of a lot of fatty acids, and we'll talk about that when we get to lipids. Myoinositol is a glucose derivative, and it forms the structural basis for many secondary messengers or signaling molecules, and again we'll talk about those later in the semester. And then ribitol, again, this ribose derivative is a constituent of vitamin riboflavin and its enzymes, coenzymes. So finally we have sugar acids. So these are carboxylic acids that are derived from aldoses, and basically the carbonyl group is oxidized to the carboxyl level. If we have oxidation at the C1 position, we have D-gluconic acid. This is resulting, the general name for this or terminology is an aldonic acid, and this specific aldonic acid is gluconic acid. If we have oxidation at the C6 position, you get the formation of an alduronic acid, and this is glucuronic acid shown here. And then we can also have oxidation at the C1 and C6 position. So this results in glucaric acid. So aldonic acids are indicated by an onic addition. Alduronic acids are denoted by uronic, and oxidation at the C1 and C6 position is indicated by the name of glucaric acid. So these reactions occur widely in biology because it is basically oxidation of glucose, which is ubiquitous in biological systems. If we look at gluconic acid, so this shown up here is naturally occurs in food, wine, and honey, and it can also be added to food as an acidity regulator. Glucuronic acid is a common building block of proteoglycans and glycerophospholipids, and makes up the extracellular matrix of many tissues. And interesting, alduronic acid, so this glucuronic acid was originally isolated from urine. So monosaccharides with free anomeric carbons are actually reducing sugar. So these molecules will reduce oxidizing agents such as peroxide, ferric cyanide, and some metals such as copper. These redox reactions convert sugars to sugar acids, the derivatives that we saw in the previous slide. And one very powerful reducing sugar is glucose, and we know that in diabetes there are high levels of blood glucose. And high levels of circulating monosaccharides gives lots of reducing power. So basically what happens is you have a lot of this reactive molecule that is present in the blood, and that results in glycosylation of proteins. And so these these glucose molecules get added to different proteins, and as you might imagine, this results in a change in biochemical properties that often translates to loss of function. And this is one of the reasons that diabetes is so such a serious problem. So this reducing property of glucose can be used to measure blood glucose levels, and actually for a long time this was how it was measured. So this is called the Fehling's reaction. So here in the Fehling's reaction we have beta D-glucose, and it interacts with copper, and this produces a red precipitate that can be measured. Now, one thing to notice is that if you look at this hemiacetal, in this form the sugar is not reactive. Okay, so it can't interact with the copper. However, this molecule is in constant equilibrium, so between the ring and the linear form. And so every time you get the formation of this linear form, you get the oxidation, or this redox reaction. So this results in a complex mixture of many 2, 3, 4, and 6 carbon acids, which I'm not really don't feel like you need to memorize the structures. But they form this red precipitate. That is what's measured. And now, of course, there's a much more sensitive way to measure blood glucose. We use an enzymatic assay to do that. So we use glucose oxidase. So glucose oxidase is an enzyme that catalyzes the reaction of glucose and oxygen to yield D-gluconodelta-lactone and hydrogen peroxide. And then these are just the structures here. Now, hydrogen peroxide is the molecule that's actually measured. And again, this is another enzymatic reaction. So a peroxidase is added, and that catalyzes a reaction that produces a colored compound. And so we can measure this spectrophotometrically. Now, one problem or one issue with this measurement is that it's a single time measurement. So it measures glucose levels at that particular point in time. And we know that glucose levels can rise and fall dramatically over the course of the day and the course of a week and so forth. And so what's really more helpful in terms of determining the severity of high glucose levels is to actually measure modified hemoglobin. And so hemoglobin is a protein that's found in red blood cells, so it's constantly exposed to any of the glucose that's in the bloodstream. Basically, there's a non-enzymatic reaction that occurs between hemoglobin, the amino group of hemoglobin, and glucose. And I'm not worried about all of these intermediates, but the important thing to note is that you get a covalent modification of hemoglobin, and we call this glycated hemoglobin. So the amount of glycated hemoglobin reflects average blood glucose levels over the lifetime of the red blood cell, which is about 180 days. And so the amount of this glycated hemoglobin is related to the amount of circulating glucose. So this modified hemoglobin can be separated from unmodified hemoglobin based on the differences in charge. Normally, glycated hemoglobin runs about 5%, and in untreated diabetics, it can be as high as 13%. So this is a reflection of long-term glucose levels. Now, what I want to point out here is that you have this glycated hemoglobin, and over time you get what is called advanced glycosylation end products, so these AGEs. Now, over time, basically what these are proteins that are modified with different sugars. You get changes in protein complexes, so you get changes in protein structure, and these AGEs cross-link with critical proteins, and they cause damage to kidneys, retinas, and cardiovascular system. And again, that's why it's so important to get these high blood glucose levels under control. So ascorbic acid is the last sugar derivative that I want to talk about. So it is vitamin C. It's a very strong reducing sugar and functions as an electron carrier. It's also an essential cofactor. It's important in collagen biosynthesis. If you don't have vitamin C, you get scurvy. It is important in metabolism of certain amino acids in the brain. Iron mobilization from spleen also depends on vitamin C. It's thought that vitamin C can prevent the toxic effects of some metals. It ameliorates allergic responses and can stimulate the immune system. And for all of these reasons, it has kind of been advertised as the magic molecule. So you'll see these pills that you can buy in the drugstore, which have very high levels of vitamin C, and they encourage you to take these in wintertime and so forth. So interestingly enough, only a few vertebrates, so man, primates, guinea pigs, fruit-eating bats, and some fish like rainbow trout and certain salmon cannot make it. So most organisms, most plants and animals can make ascorbic acid. Diabetes